Okay, today I'm going to talk about how DNA is packed into a cell. So, the packing of DNA is a huge storage issue because there's a massive amount of DNA and only a small area that you can put it in. If you think about a eukaryotic cell, like a human cell, we contain more than 6 billion base pairs of DNA, and that would measure over 2 meters long if it was stretched end to end. So, if you measure that against the nucleus, it would be 14,000 times the length of the nucleus. So it would be way longer than this, but let's just imagine that this whole structure needs to fold in on itself in order to form tiny different structures that are referred to as primary, tertiary, beads on a string. Those structures I'm going to get into in a different video, but today I just wanted you to understand how much DNA is actually packed into these tiny cells, or tiny nucleuses, sorry, inside of a tiny cell. So there are three hierarchical levels of DNA structure. The primary structure of DNA, which is just its nucleotide base pairing. So if we had a piece of DNA that was like this, and then you just have your base pairings in a linear molecule like this. Um, the secondary structure is the double-stranded helix, so that would be this shape that's coming and coiling around and spinning, and this is a terrible drawing, but if you just imagine this actually doing a full 360 turn here, and there's three different forms of this, A, B, and Z DNA shape, but once again we're not going to get into that in today's video. So the actual shape that DNA comes into once it is formed in this double helical shape is pretty important for things like gel electrophoresis and Western blotting, because the shape that it's in helps determine the size and how it'll move through the agarose gel and gel electrophoresis, which I'm going to talk about the next. This video is kind of a building block to that next video that I'm going to do. So there's three kind of main shapes that this DNA can form, and the first one is just a linear molecule. So you just had a molecule like this, which would move quite easily through a gel membrane because it's linear. So if it were to come to a part like this, it obviously wouldn't fit in this way, but if you were to rotate this onto its side, it would slide right through and it would move pretty far down a gel electrophoresis plate. So gel electrophoresis, obviously, if you were doing it for DNA, you'd have negative charges, on this side. That's a positive. So you'd have negative charge on this side, which the DNA is negative because of the phosphate groups on the phosphate backbone. So these negative charges are going to try and get as far away as possible. And the easier that the DNA molecule can migrate through this gel, the farther it's going to get. The next shape is called a relaxed circular shape. So I'll change the color for this. So that would be something that just kind of looks like this. It's just kind of like a big blob, relaxed circle. It's not really in any shape. This molecule would have the hardest time moving through a gel electrophoresis because there's really no room for it. It's really like it's spread out. It can't really get through these tiny pores that the gel makes, so it would probably move the least out of the three different shapes. The last shape is called supercoiling. So it takes place when the DNA helix is subjected to strain by being overwound or underwound. So the shape that it takes is something like this, where you have this kind of coiled up structure with all your base pairs in here. And so you can see that this one automatically, it takes a smaller diameter, which would make it kind of like a linear molecule where it can just kind of move through these pores. And there's two different types of supercoiling. There's positive, which means that the molecules are over-rotated, and underrated, which is negative supercoiling. So the way I like to think about this is like a phone cord. So when you look at your phone cord in your house, you can see that it's hanging off the wall and it's kind of coiled up like this. And like if you were to try and pull this apart, it would get into a large kind of knot and it wouldn't really pull apart that well. The way that you get this apart, if you were to try and pull it apart, like say if you were trying to replicate this uh, DNA molecule during cell division or something like that, you would use what are called topoisomerases. So the main one that you might be familiar with is DNA gyrase. So what it will do is when there gets to a point where it's really tight like this, it will form a break and then it will thread the old DNA strand through and reconnect two DNA strands so that it is completely not a knot anymore and it can move and rejoin and then continue replication with like the replication fork and the helicase following just behind that. So that's it for this video. In the next video I'm going to talk about how all of these different structures actually relate to genetics experiments like gel electrophoresis and the nitrocellulose membrane experiments.